So now that we have an idea what's happening when there's no lactose, let's see what's happening when there is lactose. So in the operon, repressor I is constitutive, which means that it's always going to be made. So here we have lac I that made our repressor. Then what happens at this point is repressor I will bind allolactose, which is a derivative from lactose, and causes a structural change in the repressor that doesn't allow binding to the OLAC operator sequence. So what that means is that we're going to have our lactose that gets bounded, and it's going to be binding to the repressor. Once we have that repressor binding to the lactose, it changes its shape. And so when it goes to bind to the operator, it actually will not bind there because that operator sequence won't recognize the shape of the repressor bounded to the lactose. So let me just make sure I include a no, which is it's not going to bind there. Okay, so then what happens? If it doesn't bind there, what is going to happen now? Well, for one, we see that the area of the DNA is now open for access, which pretty much means that now RNA polymerase is going to come. It's going to bind the PLAC promoter, which is binding here. And then when the PLAC promoter binds, then that means transcription is on, and then our genes are being made. Okay, so then that would mean our LAC-Z gene is going to be turned on, or not on, it's going to be made, and we're going to have our permease being made also. And this last bit here, it's going to be the acetyl acid transferase. No, that enzyme is also made too. Okay, but then just to sort of go back to where this comes into play, well, if we go back to our section when we talked about how this all comes together, we have our lactose outside the cell is going to come through the permease, which is made by LAC-Y. When lactose enters, beta-galactosidase enzyme is going to break that down into glucose and galactose, and then the cell will then use glucose for its energy needs. And that's what that's what our lactose is. So the main point is that with lactose, repressor I is going to bind lactose. It's going to change its shape. If it changes its shape, then it no longer can bind the operator sequence. Now that there's no repressor in the way, RNA polymerase then can bind its promoter region, and when it does that, it turns on, transcription is on, so now the mRNA is made for the LAC-Z, LAC-Y, and LAC-A. Those are then read into protein, which is beta-galactosidase, permease, and then this is acetyltransferase.